IBS Biomed Colloquium of this year. I have the great pleasure of introducing Professor Ruth Baker as today's Biomed Colloquium speaker. In a series of series of landmark studies, Dr. Baker has developed novel mathematical and computational methods and modeling frameworks for investigating developmental and cell biology. And Dr. Baker is currently a professor of applied mathematics at the Mathematical Institute of University of Oxford. And she's also a tutorial fellow in mathematics at the St. Hugh's College. And importantly, she's a Royal Society Wolfson Research Merit Award holder. Her most recent research work and the topic of her talk today is identifiability and inference in cell biology. So please join me in welcoming Dr. Baker.
Thank you, and thanks for the introduction, but thanks for the invitation. It's great to be here, and thank you in advance for listening. So I'm more than happy to take questions during the talk or afterwards. So if you want to unmute and ask a question, feel free at any point. I guess there's lots of people here that I don't know already, so I thought maybe I'd just give some introduction to the group very briefly and what we try to do. Our main focus, so we do a range of things, but our main focus is to understand how various mechanisms contribute to drive collective cell motility, so waves of cell invasion, and then how they combine with or are influenced by adhesion, proliferation, and death, and can give rise to very complicated biological processes. So kind of traditionally, if you like, in our group, we've focused within embryo development, really with a view to thinking that understanding how organisms sort of, if you like, develop in the first place gives us kind of great insights into how homeostasis is achieved and how repair is achieved, essentially. But we also focus a little bit then on areas and aspects such as wound healing and tumor growth. And increasingly over the past sort of 5 or so years, I think there's been a real shift, especially in developmental biology, in the amount of data and the types of data that we have available. So we've really seen a shift from developmental biology being a sort of qualitative discipline to one that's incredibly quickly moving towards being quantitative. And I think the real challenge is to think about how to use or to develop and use mathematical models in that context to drive new understanding. So we're trying to hit that space and connect models to data. So we try to take an interdisciplinary methodology, and both the projects I'm going to talk about today very clearly sort of sit in that space, where we have a collaboration with an experimental group who are thinking about experiments, both in wild type, but also in perturbed scenarios. And then we're trying to understand and make sense of the observations we see by using mathematical models and simulations to unravel mechanism. And what I'm going to sort of illustrate is that essentially you sort of can connect the two well by looking at various means for data analysis and model testing. And we're really thinking about working in a cycle of predict, test, refine, and predict, where all the time we're sort of essentially using the model to test hypotheses, to refine them, and to make predictions that we can test experimentally. So what are the challenges of quantitative data, you might think? So I'd argue that if you really want to kind of fully exploit the power that you have from a set of quantitative experimental data, then one of the things you want to be able to do is estimate the model parameters from that data, because then you can make quantitative predictions. And then I sort of think that that refers you back to thinking, well, is my model at the right level for the data? And a question we sort of often ask, and again, it will crop up in this talk, is whether the parameters can be identified with sufficient confidence given the data that you have. So I sort of argue that the model that's at the right level is a model where essentially those parameters can be identified, so you can be confident in making predictions in unseen scenarios. Then we think about the actual task of estimating model parameters, and importantly, thinking about what kind of level of uncertainty we have in those estimates, because then you can really think and ask questions about how uncertainty in those estimates of parameters sort of translate forward into uncertainty in the model prediction. So if I'm making predictions using my model for some unseen scenario, and I want to then go ahead and sort of test that prediction experimentally. I want to be sort of sure of the range of possible outcomes that my model might predict. Okay, so what I want to do in this talk is show how you might do that, or think about doing it for some very simple biological models. So the biology itself isn't necessarily simple, but the mathematics is. And I think that's sort of necessitated by the fact that although we've got quantitative data, there's sort of, it's still placing quite a lot of limitations on how complicated the model can be.
Right.
So we're going to think mostly about the inverse problem then in this talk, and using sort of this process of calibrating models to data to provide new insights. So I'm going to imagine that we essentially have some generally imaging data. We're going to translate that into some summary statistics of those data. So in this context, it might be sort of average density. And then essentially what we've got is the kind of a quantitative sort of description of the system. And then we're going to do parameter inference in a Bayesian framework, where we're asking whether we can estimate the probability of model parameters theta given the data d. And we write that in terms of the product of the likelihood of the data given the parameters and the prior distribution, where the prior represents anything we might know a priori about the parameters. But for much of the work that we do today, I'm going to highlight that we have little idea about what the parameter ranges could be, in particular in the second case. So we tried to take the prior as wide as possible in order to sort of capture any important details, if you like. Okay, so the first project I want to talk about was a project that was mostly carried out essentially by a PhD student in my group, Jonathan Harrison. He's now a postdoc at Warwick, and it all relates to mechanisms of RnMrna localization, robustness. So what's the idea? The idea is that in many cell types, and in particular larger cells, if you want to get proteins to the right place in the right time at the right time in a cell, then a sensible thing to do is to put the mRNAs in that place and then let them translate the protein in essentially the place where those proteins need to be, as opposed to kind of generating, having the mRNAs somewhere else, letting them generate the proteins and then sort of transporting the proteins to the right place. So we know that this process is used by many cell types to get proteins in the right place at the right time. And like I said, in particular, large cells, and you can sort of maybe hazard a guess that it could be a more sort of robust process. We know it's key in establishing the body axes for cell migration, for synaptic plasticity. And so what we want to understand is sort of what controls this process of localizing the mRNAs, putting the mRNAs in the right place at the right time, and what ensures that it's a robust process. Essentially, if a cell is going to sort of favor taking this approach, then we would assume that it's more robust than others out there. Okay, so I'm going to talk about this in the context of the Drosophila egg chamber. So this is essentially a Drosophila egg production line. So you can see about 18 strings of these maturing follicles. So if you go from the top to the bottom down one of these strings, you get a load of kind of Drosophila in the making, if you like. And they become increasingly sort of well-developed as you move down one of these strings.
Oh, so this is single egg? So is this
so this is like many Drosophila. This is called an ovariole.
I say it's many eggs.
Okay. Yeah, so there's about 18 of these strings, and they're kind of like continuous production lines. So that's a good question. So what we're going to do is just basically focus on one of them at a relatively early stage when it's only got 16 cells in it. So it's nice and simple. So we can avoid kind of, by the time you sort of get down to the bottom end, there's lots of very complicated sort of structures, but we're going to avoid all that and look at the 16 cell stage. So the 16 cell stage, you've got one cell which is actually going to become the oocyte, so become the actual organism properly. And then you've got 15 other cells which are called nurse cells, and their role is really just to support the oocyte as it develops. What's interesting about this process at this stage in this egg chamber, if you like, is that, so obviously it's got to 16 cells by a series of cell divisions, but every time sort of those cell divisions take place, they're incomplete, so the cells don't separate totally. And what you got left with is one of these intercellular bridges, and then we call them ring canals. in the field, and essentially they're important because they let mRNAs and proteins and other intracellular components move between the nerve cells and also into the oocyte. So you have this kind of very interesting picture where you can make mRNAs and proteins in the nerve cells, and then they can be transported through this network into the oocyte. And we know that there's several essentially maternal gradients of different RNAs in the oocyte, and then proteins, I guess, in the oocyte. For example, gherkin, osca, and bicoid. And we think that they're all set up by a process where essentially the mRNAs are generated in the nerve cells and transported into the oocyte, where they can translate the proteins. So we're going to focus on gherkin RNA today in terms of having the data. So this is what an actual egg chamber looks like. So this is just a single Z stack, and you can see you've got 3 of these nerve cells here, and then this one over here is the oocyte, and you can see these sort of red dots are the gherkin mRNA essentially. And you see in this case kind of already quite a lot is localized in the oocyte. And what else can you see? Oh, and you can see one of these connecting sort of tubes, these ring canals just here. This one would probably connect this cell into the oocyte. So if you go through a Z stack, you can see that really, really nicely, and you can see ring canals will pop up. There's one down here, one there, one there, and you can see all the gherkins, another one here, sort of punctate spots.
I see.
Essentially. So we're going to ask questions about how gherkin essentially localizes in the oocyte within these egg chambers. So what do we know? We know that the mRNAs are produced in these maternal nerve cells. So they're basically the mRNAs, the gherkin, for example, it's not actually produced in the oocyte at all, but it's just produced in these surrounding 15 cells. Mm-hmm. And then we know it's transported to the oocyte through this sort of network of ring canals. So we thought about a range of different models, and this was by no means the first model that we tried for this system. But actually, in the end, it was the simplest model, but it was the one that was, I think, most relevant. So we had quite a simple compartment-based model where all we're going to do is track. So this is our network of 16 cells. It's reliably generated. It always takes the same configuration. We're just going to track the number of mRNA complexes that you see in each one of these cells. So cell number 1 is the oocyte, and then the nerve cells are the ones in black, labeled again in a very stereotypical, yes. So in a consistent pattern. And so what I said, one thing I was going to say was when these mRNAs are transported between the nerve cells and into the oocyte, they're packaged together so that in one kind of those bright spots, you might see multiple mRNAs together with some proteins that can be transported in the sort of along the network in the cell. So we've got a compartment-based model. We've got 16 equations, 16 ODEs essentially, that track the number of mRNA complexes in each cell. And then we're going to assume that we've got bias transport between these complexes between cells if they're connected by a ring canal, so if they're linked in this network. And then we've got production of the RNAs in each cell. Mm-hmm. Okay, so that leads to a system of 16 ODEs. You essentially kind of wind up with 3 parameters. You've got a parameter that controls production rate, one which controls the transport rate, and a parameter nu hidden inside this matrix B. So the matrix B, the details don't really matter too much, but essentially they just encode the network structure. And then the parameter nu encodes this bias in the transport. In the field, strangely, you always take the bias as being away from the oocyte. So we're going to anticipate ending up with small values of nu when we try and do calibration of the model.
So every node has the same production rate here?
Yeah, I mean, in reality, maybe there's fluctuations slightly, but simplest model, and we did check this, but yeah, the simplest model is that every node has the same production rate. And actually, this vector V just encodes the fact that there's no production in actually cell number 1.
I see. Except for the first one.
Yeah, except for the first one, because we know there's nothing there.
Got it.
So this model's nice because you can write down an analytic solution, which is great, and then you can do lots with it to estimate model parameters. So the first thing you can do as well, I guess, if you want to, is just look at typical behavior of the solution for different parameter values, and this is the kind of thing that you might get. So we're always going to plot, so this is not a continuous plot, we just join the dots as it were, but we're just plotting the number of mRNA complexes in each of these nerve cells labeled according to these numbers in the network. So yeah, parameter inference in a Bayesian framework. And in order to do that for this model, we need a measurement model. So we're going to have a negative binomial measurement model with parameter sigma. And then this vector phi in here actually just introduces an additional parameter, lowercase phi, into the model. And that's important because going back to here, we know, I said before that several mRNAs were packaged up into a kind of a complicated, into a complex essentially for transport. But we know that when they're actually kind of transported on the final bit of their journey from one of these nerve cells surrounding the oocyte into the oocyte, they're kind of repackaged to put more mRNAs into one of those complexes. So when you see a bright dot or when you see a dot of expression in the oocyte, there's actually more mRNAs in there than there is in one of the nerve cell red dots. So that was a bit of a complicating factor. And we just
Can I ask a question?
Yeah.
Sorry for interruption. In the compartment model, is there any driving force toward the oocyte, number one, or the mRNA is trapped in the number one compartment?
So there's a bias transport, right? So essentially the transport is biased away from the oocyte, if you like. So this transport parameter nu will lie between 0.5. And that will represent how biased, what kind of, if you think about it in probabilities, the chance that a molecule will be transported towards the oocyte versus away from it, basically. So that represents the transport. So when things get into the oocyte, they're not transported, they're not stuck there, they can actually come back out. And that's all encoded in this matrix here. You can see the terms up here.
Okay, thank you.
Yeah, so we've got now, I think that the difficult thing is that we've got now 5 parameters. So we had A and B and nu from the original ODE model, and then we've got sigma and phi that come from essentially sort of kind of experimental aspects, if you like. So the measurement noise and also this higher order assembly. So the thing that goes wrong, if you like, is that as soon as you kind of put in the details of this higher order assembly process, the model becomes unidentifiable. So you can't sort of essentially uniquely determine each of the parameters or confidently estimate each of the parameters in the model on their own. So our solution to this was to try and essentially get at this complex assembly parameter on its own first. So what we did was, again, this time the gherkins in sort of purple, magenta, is we looked within the nerve cells, we looked in the oocyte and we looked at the background, and we essentially quantified the fluorescence intensity of the spots, essentially the expression, both within the oocyte, the nerve cells in the background, and used that to estimate that assembly parameter first. So here is essentially the sort of normalized intensity sort of plots for backgrounds within the nerve cell and within the oocyte. And we could use that to estimate this parameter phi. So we think it's got a median value of around 0.4. So somewhere between 3 and 4 mRNAs are packaged together, moving from the nerve cells next to the oocyte into the oocyte. And then we can get a credible interval on that too. So the good thing about that is you can use these results to specify a very strong prior for phi in the full model. So that essentially alleviates those identifiability issues because you've managed to relatively confidently estimate one of the model parameters using other experimental data. So then you can go back to the model and think about parameters, getting a handle on the rest of the parameters. And one of the things that you can do nicely is think about sort of, if you like, the long time behavior of the model. So essentially at large times, the solution is just growing linearly in time. So you can forget about what's going on with the first 2 terms in the model and just see that essentially the distribution of mRNAs across these, across the nerve cells and the oocyte will all be governed by this vector K2, which you can get a handle on analytically. So essentially, if you kind of normalize everything to the distribution in the oocyte, then this parameter, this vector K2, will allow you to nail down the parameter nu, the bias parameter. So essentially, what I'm saying is that you can ignore the kind of rate constants that relate to production and transport, and get a handle on you by looking at this kind of longer time data. And when you do that, so this is the prior distribution, essentially not putting much weight anywhere. In particular, on the in parameter space, then you can see that the posterior distribution is quite tightly peaked around, I guess, 0.0, I don't know, 4, 5-ish. Certainly, basically giving a strong indication that this transport bias parameter is very small. And so I said that the bias was always away from the oocyte. So that means that on average, everything is being funneled towards the oocyte. So it's a bit back to front, but it kind of makes sense, if you like. So once you've got that, one of the things you can do is do a posterior predictive check. So essentially what you want to do is sample from that posterior distribution, simulate the model, and check that the data you have is essentially in line with the model. And it appears to be the case. And then you can go forward and you can tackle the full dynamic regime to get a handle on those parameters A and B. And the short kind of answer here is that you can get fairly good estimates or fairly confident estimates of both A and B. The posteriors are relatively nicely peaked. And then if you go back and you think about the model, you can see that in terms of, where's the model gone? In terms of the relative sizes of these terms in the model, so production versus transport, you essentially those posterior distributions tell you that the production rate is of the same order of magnitude as the transport rate. So it tells you that essentially production and transport are relatively carefully balanced in this term, so in the system. So it's not that one or the other one of them is essentially appears to be rate limiting. You sort of need both of them. I rely on both. Okay, and again, you can do this kind of posterior predictive check where you sample parameters from your posterior distribution, simulate the model, and then the range of outputs of the model are in gray and the experimental data are the dots. And I've arranged things here slightly differently than before, where on the far right-hand side, this is cell number 1, which is the oocyte. And then the next kind of column going towards the left are cells 2, 3, 5, and 9. So they're all the cells that are kind of a distance 1 on this network away from the oocyte. And then you get all the cells at a distance 2 away, 3 away, and 4 away. And that'll become important in just a minute. So I guess the take-home message really is that the model seems to be doing a relatively decent job at predicting the data. So this is all well and good. But,
so Ruth, what is the meaning of these columns here? What is the difference?
So essentially, yeah, just a way of arranging the data. So these are all the cells that are a distance 1 from the oocyte. So these are cells 2, 3, 5, and 9.
Oh, 2, 3, 5, I see. Just yeah, the 1. I see.
Yeah, so these are all the cells that are a distance 2 away. So it would be like 4, 6, 10. And it's just important later, actually. So at the moment, it probably doesn't make that much sense, or it's not obvious why you would want to look at this, but hopefully I'll convince you in a minute that it's useful.
Wow. Because the most right-sided one has really large scale.
Yeah, so the scales are definitely different because it's all accumulating there, I think, just because this bias term is so big. But yeah, good point. What was I going to say? Yeah, so I guess, it's great that the model's working, but, in biology, we kind of learn the most where in the context where we kind of challenge our model and there's something wrong with the model. Because if there's something wrong with your model, you just know that there's something that you haven't quite understood properly, and you can go and search for what that might be. So we thought that we would try and challenge our model to predict the behavior of an overexpression mutant. And the idea here is that you aim to engineer your mutant genetically so that you get 2 copies of this gherkin gene in every cell. So the idea is that if you have 2 copies of the gherkin gene in every cell, so this is a picture over here on the right, you can see there's much more, there's much more gherkin right in these nurse cells. Then the simple model extension would be to say, okay, I'm going to use the model that I had before with the parameter values I had before, and I'm just going to put a 2 in front of the production rate. So multiply the production rate by 2. And if I do that, hopefully this model can predict the dynamics of this overexpression mutant. So we challenged the model to see if that was the case. And these are the results. So again, the gray is kind of the range of possible predictions from the model. And the red dots are the experimental data. And hopefully I'm going to convince you that it's not doing a bad job. We're kind of getting quite good estimates for the everything kind of quite close to the oocyte. But if you look further away from the oocyte, then we're not, things aren't so great, right? It's not doing very well. So you can see that the model's really, so these are the model kind of envelope of the model results. They're really under predicting essentially the amount of mRNAs that you're going to get in those cells. So something's not quite right. You can also look at the data and you can ask a question about, okay, we're essentially generating twice as much mRNA in each of these nurse cells, or we're generating at twice the rate. So in our model, kind of by extension, you'd be accumulating mRNAs at twice the rate. But when you eyeball the data, this is the number of mRNA complexes at a given time. You can see that in the wild type, you're kind of accumulating at some rates indicated by this sort of blue-green line and dots. Whereas at the overexpression mutant is not essentially accumulating mRNA in the oocyte any quicker. So despite the fact that we're pumping in twice as much mRNA or twice as much gherkin into the system, somehow it's buffering against that to localize the same amount, right, of mRNA over time. So that's quite interesting. This is kind of a real kind of indication that actually this process is very robust. And something in there is buffering again, you know, something in there is driving this or causing this robustness, and it's not in our model. So we need to think about how we could adapt the model to sort of become robust essentially, or display robustness. So we thought for quite a long time, and there was a really long list of things initially that, or mechanisms that could be at play that would allow the system to be robust or give it robustness. So the first thing you might assume is that if you're kind of generating these mRNAs at a really high rate, then essentially there's blocking at the ring canals. So just in the same way as, you know, if you go to watch a football game, a soccer game, and you're trying to leave the stadium at the end, there's lots of kind of queuing, right, around the exits because they're small compared to the number of people, then maybe this kind of idea is what's happening in Drosophila as well. So you get blocking. Going back to the question from earlier, which was a really good question, we thought maybe there's some kind of inhomogeneity in the production rate. Now, there is almost certainly a bit anyway, but in this context, we weren't necessarily confident that we'd manage to get 2 copies of the gherkin gene into every nurse cell. So we essentially allowed for the fact that there could be variability in the production rates across all of the nurse cells. And then we also sort of hypothesized that there could be an impact from density dependent transport. So essentially these gherkin sort of mRNA complexes, we think are transported by the kind of microtubule network of the cell. And so maybe there's just not the motors around. Essentially what we're doing is saturating that transport process. It just becomes overloaded. And so there's a limit to how much it can transport. So the idea that we had was to sort of represent these different hypotheses as a collection of models, so all combinations of them, and then use some model comparison approaches to sort of essentially see or evaluate their plausibility given the data that we have. So which one of them is most consistent with the data effectively. So you wind up with 7 models. So model 0 is the base model where you just multiply the production rate by 2. And then models 1, 2, and 3 are just the different hypotheses that I just described, or include them. And then 4, 5, and 6, they have 2 of those kind of hypothetical mechanisms included, in model 7 has all of them. So by rights, model 7, it's got the most flexibility, maybe that can fit the data well. So we used a couple of different approaches to compare the models. But in both of these approaches, it was model 1 that was favored the most strongly. So model 1 encodes the hypothesis that there's blocking at the ring canals. So essentially saying that it's like this kind of crowding effect where people are trying to leave a stadium or after a sports game or after a concert. And essentially it's that sort of blocking of the ring canals that's stopping the mRNAs accumulating too quickly in the oocyte.
So what is the method here you used? How did you?
So this is so this essentially evaluates the plausibility of the different models by weighting in different ways. And I've got a slide at the end that sort of talks about that a bit more.
I see.
But the take-home message here is that in these BMA weights, you assume that in the set of models that you're testing, that one of them is the correct model. And so in this case, we get loads of weight on model 1, whereas in this stacking weights approach, you don't make that assumption. So you kind of essentially kind of understand that the correct model might not be in the list of models you've got. And so it's a bit more conservative. And so there's a bit less weight on each of them, I'd say. Sorry, on model 1. And you can see that there's also some support for models 3 and 5. But in particular, so what is it? Model 5, I think, is next best weighted. And you can see that's a combination of blocking of ring canals and inhomogeneity and production rates. And that is almost certainly true. It goes back to that question right at the start. There's almost certainly some inhomogeneity in production rates. And actually mechanism 3 on its own is relates to this inhomogeneity of production. So this is all kind of consistent, I think.
I see. Because
So
interesting
Ruth,
that
I have a
M7
question.
has the most flexibility, but wow, the M7.
Yeah, well, I guess that you kind of take that into account in the evaluation. But I think it's interesting that you can essentially kind of highlight just one of these mechanisms as being sort of maybe the most important, and then we sort of know there's going to be inhomogeneities in the rates at which we're producing mRNAs. Yeah.
So I have a question. Yeah, I think blocking is one kind of density dependent transport, because if there are many higher density, then the ring canal might be blocked, which is another some special case of the density dependent transport.
Yeah, so I guess with the blocking, we're sort of we're assuming that it's blocking because that channel becomes blocked. And I guess with the density dependent transport, we were thinking more about this coming from this sort of transport network of the cell being saturated. So kind of assuming that with people, we're transporting everyone on buses, and at some stage there are too many people to fit on the bus. So you can't transport anymore, right? You're kind of overloading the system in terms of the transport, and this is to do with that blocking. So they're very this is a very, very coarse-grained model, so it's difficult to kind of really tease apart the difference. But
okay,
yeah, I think I would assume that one would come under blocking a bit more, whereas this density dependent transport is letting the production rate essentially is modeled as a Hill function, so it saturates at some point.
I got you. Thank you.
Yeah, I think to really think about it in detail, and when we tried this, but I think we need better data, then we would want to develop quite a detailed model of maybe like a single nurse cell and look at the transport network and really try to evaluate kind of what was the limiting factor in transport. But again, yeah, I think really good question. So yeah, one thing I wanted to say is that you can actually, if you go into some of the images we had and you sort of zoom in a bit, you can see this sort of buildup of mRNAs around the ring canal, which is about here. So I think that sort of lends support to the fact that there is some kind of blocking. And obviously the kind of the thing we should do now is go back and sort of probe that and test that a bit further. And as I was saying, that maybe building some more detailed models surrounding those the transport behavior. But I'm going to sort of conclude this bit of the talk, because I'm going to kind of switch gears in a minute and say that I think one of the strengths of this bit of work was that the fact that we took a very, very simple model, but it was really the kind of connection with the data that made it powerful and distinguishing between hypotheses. So if we hadn't have been able to calibrate the model to data, essentially, we couldn't have done this sort of model comparison in a formal way, and it would have been quite difficult to sort of distinguish between those different models. But we think in this context of mRNA localization, and in particular gherkin and Drosophila, that's a tightly regulated balance between production and transport, and that we think this crowding of the ring canals helps to regulate the process and make it robust. So there's definitely more work to do, but that's the kind of where we're at to at the moment. Are there any? I'm happy to take more questions on this part of the talk if anyone has any.
I have a question.
Yeah.
So, is there specific reasons that the compartmental model doesn't have a cycle there?
In what sense, go on, sorry.
Oh, you mean the cycle in the network structure?
Yes, I saw that there is no cycles between cells.
Yeah, so just because essentially this came from a series of cell divisions. So sorry, I'm going to flick up through the slides. Because it came through a series of cell divisions, there's essentially no room for there to be cycles in the network. So kind of one cell divided into two, and then these two divided, etc. And so that's what generates that network. But yeah, good question.
Okay.
Sorry, I missed
Oh,
that.
sorry, it's my phone. Sorry. Yeah. Okay. So I'm going to switch gears totally now and move up to the level of the kind of the cell, not thinking anymore about what's going on within cells, but thinking about single cells and their motile behavior and how that might be influenced by electric fields. So totally different. So what do we know? We know that there are sort of lots of different types of environmental cues that cells can respond to. And we can use them in development. They're often used to direct cell motility. So maybe the most obvious one is chemotaxis. So we have some diffusible chemical. This is what's happening in the movie. The chemical diffuses from some localized source, decays, that sets up a gradient, and the cells move up that gradient towards the source. But we also know that there are other mechanisms like haptotaxis. So this would be gradients in, for example, adhesion sites or substrate bound chemicals, and durotaxis is gradients in essentially sort of the stiffness of the extracellular matrix or the rigidity of the environment. So we've known for a long time about these, and probably chemotaxis is the most kind of well modeled mathematically. We're interested in a sort of a cue again that has, I think, been known about for a long time, but not really been that well studied or modeled, and that's electrotaxis. So this is a directed motion of cells in an applied electric field. So on the left, you can see very quick, sort of clearly this population of cells, which when the field is directed left, right, move towards the right. And when it's up, down, up, they switch and they move up. And you can see that kind of very quick response. Right. And then in the movie over here on the right, you can see a similar thing where the field is essentially switched on after a certain amount of time and they all move down. Okay, so electrotaxis. Why are we interested in electrotaxis? Well, we know that a huge range of cell types can undergo electrotaxis, and we know that there is kind of electrotaxis takes place kind of naturally, if you like. So if you have a skin wound, then there's gradients of sort of there's an electric field generated within that wound, and that can potentially help wound healing. So we know that it's sort of prevalent, and we know that lots of cells can respond to electric fields. And so I think for me, one of the nice things about electrotaxis is it's got then potential as a bioengineering tool. So compared to sort of chemotactic gradients, electric fields are relatively easy to set up, to control, and to manipulate over time and to keep stable. So maybe they offer the potential as a bioengineering tool in the way that these other kind of methods of controlling cell motility don't. Cool. So our sort of eventual goal is thinking about turning electrotaxis into a bioengineering tool and you know, moving in the direction of regenerative medicine and wound healing. Because if we know there are essentially endogenous electric fields in wound healing, can we exploit them? You could ask whether we could exploit them to promote or accelerate wound healing. So in terms of the mathematical approach, right, there's been loads and loads of models out there by, you know, a huge number of different researchers thinking about the motile behavior of single cells without an electric field. And then there's been lots of models as well that have looked at the motile behavior of cellular collectives without a field. And in some of them as well, there's essentially a scaling up from one to the other, right? So that there's a kind of formal link between the two. So as a start, our idea is to think about how we can take models of single cell motility in the absence of an electric field and ask how we might adapt them or extend them to replicate or to describe the motile behavior of single cells in the presence of an electric field. And the kind of long-term goal, right, is to is to be able to predict the behavior of a collective, of a population of cells in an applied electric field. But we're going to focus simple for now and look at single cell behavior. Okay, so this is the data that we have for our experiments. So this is all from Minshall's lab at UC Davis. And essentially what you can see is we've got two replicates of an experiment where here you've got human corneal epithelial cells and they're just essentially wandering fairly randomly within the dish. So there's no applied electric field. So we image them about every 5 minutes for 5 hours, and then we can collect tracks of cells from that. And then we've got some data also under an applied electric field where for the first hour, there's no field. And then you switch the field on 2 hours from left to right and 1 hour, 2 hours from right to left. So you can see the field goes on in this direction and then it's reversed and the cells all move back again. So we've got, in each case, we've got same cell type, we've got 2 replicates, and we can collect behavior on the cell trajectories over the course of the experiment. And we want to ask the question whether we can write down a model that allows us to understand how the electric field is impacting the motile behavior of the cell. So the framework that we took was, like I said before, to start from what I describe as an autonomous model. So this is a model of a cell motility in the absence of a field, and then try to extend it. So our autonomous model is essentially an SDE-based model. So we're going to update position based on the cell having a velocity v. And then what we're actually going to model is the polarity. So we're going to write v, the velocity as being the product of the modal cell speed scalar v and the cell polarity p, vector p. And we're going to write down an SDE for how that describes how the cell polarity evolves over time. So we've got a kind of a sort of drift component to that, so a noise component to it, and then we've got a sort of advective component in there as well. So we're assuming that essentially the polarity is a random walk in some potential W. So then the first thing we did was to think about kind of, if you like, eyeballing the data to try and understand what a sensible form might be. Sorry, that's annoying. Sensible form might be for this potential W. So if we look at our control data sets, then we can, each one of these lines is one cell over the course of the experiment. And you can look at the distribution of displacement distances over that 5 hours. So doing it for individual cells shows you that cells kind of spontaneously repolarize and move with some positive speed, but they can spontaneously sort of depolarize, right, and then reorient and move off again. If you look at the kind of evolution of the direction autocorrelation, we see that that decays over time, consistent with the fact that there's kind of cells are undergoing some, the polarity is essentially undergoing a random walk, if you like. So then the model that we wrote down essentially, it becomes thinking about kind of the form that this potential function W should take. So we know that when there's no Q around, that the direction in which cells polarize is unbiased. So in fact, W, instead of being a function vector P, is just a function of scalar P, which is essentially the magnitude of the polarity. And then we write down W as taking this functional form. And so this really, all this sort of encodes is the fact that cells are going to spontaneously, cells will polarize, and then they'll have a sort of they'll kind of essentially be undirected in terms of their the direction of their polarity, and they'll tend to sit around a value of P equals 1, right? There's a potential well there. Okay, so this is the model that we were working with, and we've got 3 parameters in the model. So we've got a timescale parameter D and a potential energy barrier delta W. So this is beta over 4, where beta kind of sits inside this potential function. And so they control the rate at which the cells transiently depolarize. And then you've got a third parameter V, which is the modal cell speed. Okay, so we're after kind of essentially estimating these three parameters from the data. So the way in which we did that was to take, again, a Bayesian approach. So we want essentially to estimate these three parameters using the data, which is these cell tracks in the absence of a field. And we write that again as proportional to the product of the likelihood and the prior. And this time we can split it into the individual likelihoods of each of those cell tracks we observe. So the likelihood, as with many of these models, actually is intractable. So we decided to use a method called synthetic Bayes. So the idea here is you're going to define some summary statistics of those trajectories. So it could be things like the total kind of X displacement over the course of the experiment, or the total distance traveled, or the Y displacement, etc. So you take these summary statistics, and across your data, they've got some distribution. And then you're going to approximate the likelihood with the synthetic likelihood of this data. And you're going to assume that there's the likelihood of the summarized data under the assumption that the likelihood is Gaussian distributed.
So basically,
so
you're using the just mean, I guess, mean and variance.
Basically, yeah, you're looking at the parameterizing this by the mean and the variance of these summary statistics.
Mm-hmm.
And so it becomes a much more tractable problem then from a computational point of view, because you've got a tractable likelihood. Yeah. So then here's the posterior distributions. So on the diagonal, what I've shown is the posterior distributions, the marginal posterior distributions for each of the parameters separately. So this is the cell speed, the energy barrier, and the rate constant D. And I think I'd argue that they're relatively confidently estimated, right? Each parameter is identifiable. And especially I think that comes from if you look at these plots underneath the diagonal. So these axes show you the range of the prior distribution. So we had no idea about what these parameter values should take. Some of them aren't kind of physical, so we couldn't have any kind of guess. So we took very wide prior distributions, and you can see that the posterior mass is really concentrated. I mean, down here, it's in a very tiny region of that sort of prior space, which tells you that really we're focusing on a kind of very narrow range of parameters that are consistent with our data. And in the top, we're just showing you those posterior distributions zoomed in a bit. So what can you do with the model? Then you can obviously make some predictions. So you can look at the simulated positions of cells by drawing parameters in the model and compare them with observed trajectories. So here I'm just translating every cell to be the origin at the start, and then that makes the comparison a bit easier. And I can also make some predictions of cell characteristics, and they are kind of predictions of characteristics that I couldn't easily estimate from an experiment, right? So what's the probability, for example, that a cell is polarized at any time? So the nice thing about having calibrated the model is I can make these predictions even though I can't sort of get at those quantities from the data. So then we wanted to think about what the electric field would do in terms of perturbing or impacting the cell motility. So we've got an applied electric field. So we're going to call it U. And so there's no field in our experiments for the first hour. Then it's directed to the right for hours 2 and 3, and to the left for hours 4 and 5. And our question is, in the context of our model, how does this applied field impact cell motility? And this, if you recall, was the model that we were working with. So the idea we came up with is to think about all the different ways in which we could encode and sort of a change in motility caused by the electric field in this model. And we came up with 4 different, if you like, electrotactic effects. The first one we called the velocity bias. So this is the idea that the velocity of the cell is its velocity kind of in the absence of a field, plus an additional term which represents the fact that we assume that the field puts an additional force on the cell. So it's in the direction of the electric field, and it's got strength gamma 1 relative to the sort of the no field velocity. Secondly, we thought that it could cause the cells just to increase their speed. So it doesn't matter the direction in which the field is in space. Okay, it just says that cells increase their speed by a factor of gamma 2 in the presence of an electric field. So it's a sort of chemokinesis idea. And the next one would be speed alignment. So this says that cells change their speed by a factor gamma 3, and the extent to which it changes depends on the alignment between the polarity of the cell. So this is a unit vector in the direction of the cell polarity and the field. So if the polarity and the fields are aligned, then there's an increase by a factor of gamma 3 in the speed. If they're totally in opposite directions, then it decreases by a factor of gamma 3, and then the whole sort of spectrum in between. And then the last one is a little bit different in terms of the model. This encodes a polarity bias, which says that essentially cells are at rate gamma 4 relative to the kind of wild type case, if you like, or no field case. At a rate gamma 4, they try to align their polarity with the field direction. So the idea is to think about all the different possible combinations of these different electrotactic effects and compare, essentially do what we did before, do a kind of model selection problem where we compare the ability of each of the models to replicate the data. So we are going to do this again in the same framework as before, but instead of just having 3 parameters before, now we've got up to 7. But we can still do Bayesian inference in this in the same way as before. So we're still going to use synthetic Bayes. And now we've got sort of a range of data that we can use. We can still use the data in the absence of an electric field, because that's relevant if we set these gamma parameters to 0. And we're going to use the data with the field switched on, but I'm only going to use some of it. So I'm going to use the first 3 hours of those experiments with the electric field switched on. So it's 60 minutes where there's no field, and then 120 minutes when the field is directed towards the right. And then I'm going to use the final 2 hours as a test of my model. So just as a reminder, I'm just going to use this data where the cells all start moving towards the right in my calibration process. So what do I have? I have a collection of 16 different possible models. So these are all combinations of these different electrotactic effects. So I've indexed them 1, 2, 3, and 4. And then I can write down an objective function, which is essentially the log of this quantity px, where essentially px sort of evaluates the ability of the model to give rise to the data, if you like. So it's the integral over theta of the product of the likelihood and the prior. So it's essentially the evidence for provides you with the evidence for model X, if you like. So we've got 16 different models. So we basically repeat the process of calculating this quantity J for each of those 16 models. So we have to essentially evaluate the likelihood, integrate that, and that gives us the evidence. And here's the ranking. So essentially, longer blue bars mean more support for the model. And what you can see is that a model that incorporates only electrotactic effect number 4 is essentially the model that's got the most support lent to it. Okay, and actually you would think that a model that incorporated kind of all of the different effects might do better, but it doesn't. It's quite a long way down the list. So it really is model 4 that's kind of most consistent with the data, or best supported by the data. What was that model 4? This was the hypothesis that the bias in cell motility came from the fact that the cells were preferentially polarizing in the direction of the field. So it's this idea that we add an additional term into the polarity evolution equation to represent alignment with the electric field. Again, I can show you the posterior distribution in this case. Hopefully can convince you that we've got relatively good and tight estimates of the different parameters, especially when we compare them to the support of the prior distribution. And then I can do more. I didn't talk about this for the first kind of for the autonomous model, but I can do a posterior predictive check again. I can essentially simulate from my model by drawing parameters from the posterior distributions, and I compare the model output, which is in blue, with the actual observed data, which is in black. And I want to do this initially in summary statistics space. So these 4 summary statistics, looking at horizontal displacement, overall displacement, path length, and interval variability, they're the 4 summary statistics that I used in the synthetic Bayes approach to calibrate the model to the data. So I'm doing the check initially in that data space, or in that summary statistics space. But I can also essentially compare the predictions of my model with the data in terms of the trajectories that I see. So on the top row, we've got essentially the data and the simulation results in the absence of an electric field, and above we've got the data. Sorry, and below we've got the data and the simulation results in the presence of an electric field. And they might not be perfect, but it's doing a reasonable job. So I'm nearly finished, I promise. But lastly, what I wanted to just show you was that then we can make some predictions for the test data. So how does this model that we've calibrated do when it comes to predicting some data that it hasn't seen? So this was this final 2 hours of the experiment. I haven't used that data yet in calibrating the model. So I want to understand how well the model can predict those data. If I look in summary statistics space, it all looks fairly good. And it doesn't look too bad when I look at it in terms of the observed and the predicted trajectory. So after 3 hours of the experiment, we translate the position of every cell to the origin, and we look as it essentially reorientates. So it was moving towards the right. The cell is going to reorientate and come back to move towards the left. And you can see that that happens in both in the data and in the model simulations. But it's definitely the case that in the experiments, the cells tend to reorientate much more quickly than they do in our model. And why is that? I think there's a bit more work to do here, and I think it's probably for 2 reasons. Number one, that we've done all our inference using summary statistics, and it's quite possible that the summary statistics that we've used haven't really captured the reorientation process quite accurately enough. So something's lost in the kind of reduction of the data dimension. And it's also probably a bit to do with the fact that we've basically taken data from experiments. If I can just hop back up, sorry. Here we go. We've taken data from experiments that, whilst it's a low density, it's not a kind of extremely low density. And you can certainly see that as the field direction is switched, so about now, that you can see that there's interactions between the cells that might impact their motility. So I think it could also be kind of essentially the fact that the model's not quite accurate enough in the data, but that certainly gives us something to work on in the future. So as a take-home message from this bit of the talk, we're sort of predicting that the observed bias in cell motility is coming from the fact that cells want to polarize in the direction of the field. I like this framework because we can very easily pick it up and apply it to other experimental scenarios and different cell types. So some cells, when you put them in an electric field, move towards the anode, some move towards the cathode. There's certainly differences in cell behaviors in the same field. So it'd be interesting, I think, to look at that across different cell types. But I also think it would be interesting to see if we repeated the experiments at a range of different field strengths, whether you always pull out the same mechanism and how essentially the strength of the field relates to the strength of the effect that you see. And then in the future, so we're working on this at the moment, we're extending the model to populations of cells. And I guess the very long-term goal would be to be able to use the model to say, okay, if I want to see, move my cell population from here to here along a desired trajectory, could I develop an optimal electric field stimulation protocol in order to do that? So that comes back to sort of optimal control ideas that I think someone mentioned earlier. So as a summary from the whole thing, hopefully you've demonstrated that if you want to exploit quantitative data, you want to be able to calibrate your model to those data. And that this has allowed us in these cases to understand which biological mechanisms are important in driving the system dynamics. And we definitely wouldn't have been able to do that without a quantitative comparison. But I will say that I think determining the right model can be really, really difficult. So certainly we found that with the mRNA localization model, it wasn't an easy thing to do, to figure out what model was right. But the analysis of model identifiability can really be helpful during the model building process. And I think a Bayesian approach that allows you to quantify the uncertainty in your parameter estimates and then in your model predictions is really useful. So I'll stop there. Happy to take questions. Just like to thank Tom Prescott, who did the electrotaxis work when he was a postdoc in my group in Oxford. He's now at the Turing Institute. And the data and the experimental collaborators were Min Zhao and Kan Zhu at UC Davis. And then the mRNA localization project was carried out by Jonathan Harrison when he was a PhD student. And that was in collaboration with Elan Davis's lab and in particular Richard Parton in Oxford. So thanks for listening, and I'm happy to take any questions.
Thanks for the great talk, Ruth. So from now, let's have a Q&A session. So if you have any questions, please unmute yourself and ask a question to the speaker. Or if you cannot unmute, just leave the question in the chatting room, then I will read it for you. Anyone?
So,
okay.
yeah. So I want to ask you a question about
maybe you can introduce yourself first and then
okay.
Yeah, anyway.
Okay. By the way, my name is Seokju, and I'm working at Professor J, especially on intracellular dynamics. So how do we can reduce the spatial heterogeneity of intracellular events? And like other aspect, my research is finding key factors of cancer immunity using immune cells. So I'm trying to find which factor is key factor of immune response against cancer. So I was really interested in the part where you applied pseudo BMA or stacking to quantify the plausibility. So can I ask you the relation between stacking and Bayesian information criterion? So like, lots of information criteria are used in
so
statistics.
Effectively, we're essentially looking at the models. So if I've got some unknown I want to make predictions about future behavior. So really like behavior data that the model hasn't seen, if you like. So I'm going to try and ask how well does the model capture some data that it hasn't seen based on it's being calibrated to a given data set Y using, and this is the index of the model, if you like. So essentially you're kind of thinking about this, taking this approach, which is I think a little bit different to BIC. But I can send you the reference that we used to do that. But certainly I think one thing that was sensible to do was to look at, and I think I mentioned this in the talk, was to look at different ways of weighting each model. And certainly when we use the BMA weights, then we got this really, really strong support. So I think if I do this, you might not be able to see anymore, which is, hang on, let me go back instead of going through. So certainly it was the case that if we use the BMA weights, which assumed that let me share screen again, that the correct model was in the set of models that we had, we got maybe kind of quite artificially strong support for this model 1 over all of the rest. Whereas when we relax that assumption to kind of acknowledge the fact that the true model isn't in the collection, by virtue of it being a model isn't in that set, then we can maybe get some, I'm not saying I'm going to say sort of slightly more reliable results, but at least what we see is that we still got strong support for that model. I'd hope that would be the case. But then we also introduced, so if I look at models 3 and 5, they also bring in this inhomogeneity in the production. And I think that probably is quite important, right? So we know we sort of know that there's probably an inhomogeneity there. So it was, I think, kind of looking at different ways of weighting models and interpreting that is important in that context.
Okay, so so in calculating such kind of weight or plausibility, so are the calculation or algorithm to obtain them is related to the way calculating, like, like well-known scores such as like Bayesian information criterion or like Kaiki information criterion? Like, are they directly related or not?
Do you know offhand? I couldn't tell you. I'd have to go back and think about it a bit more. But it's a good question, and I'll have to think. And maybe we can follow up on that one. That's good question.
Okay.
So thank you, Ruth. It was an interesting talk. My question is, when you evaluate model, is there any penalty about number of model you consider? Because I thought considering only model 4, for example, is a special case of considering all the model. And if we make the gamma 1, 2, 3 are small, like a 0 or almost 0, that is almost the same as considering only model 4.
Yeah, so we certainly looked at, let me find the slide. So we certainly looked at, here you go. Let me share again. So we certainly looked at all combinations. So if you look down here, this is what happens when you look at evaluating, including all of the different models in there. And so actually there wasn't more support for that, right? So even if so I don't know offhand what the values of those gamma 1, 2, and 3 were, but certainly even adding them as relatively small parameters didn't improve things, right, compared to just having model 4. I think that's because implicitly you are penalizing kind of the more complicated models through this integral.
Oh, I see. Okay.
It's sort of in there. There is essentially a kind of subtle. So it wouldn't be like having So I think that's why that partly happens.
Got it. Oh, nice. Thank you.
Any other questions, please? I think, So any question from the audience to the Ruth? So I see. So Ruth, so for that model selection method, I guess if we go to your reference, can we find a detail about that method, I guess?
Yeah, so in both of them, the references are there, or I can send them to you. That's no problem.
Yeah, because we also do a lot of model selection, but we feel that those methods looks very promising for us. So, those two methods are your preferred method, or are there any other options you usually use? Or
so I think it's important. We've looked at a range of different approaches, but like if you think about the different information criteria, I think if you've got a given model and some data, then different information criteria might actually favor different models. So one of the things that I think is quite important to do is evaluate some different ways of maybe for a problem of model selection. But yeah, I mean, this is in a way something I'm like not totally an expert on, so we're sort of learning in the group as well in terms of thinking about what the best approach is and the best way to do things is. I think it depends. I think it sort of depends a little bit on the data that we've had, and sometimes as much as anything, it's been about learning about different approaches and what they can tell you. So we've often tried a number of approaches. But I think that's sort of sensible. I'm not sure I have any expert answers.
I see. Okay, so maybe. So any other questions? If so, maybe. But before finishing today's session, maybe the other member you haven't talked with Ruth, maybe you can introduce yourself briefly to her before we sending her. Maybe you mean you can begin?
Okay, hi Professor. My name is Min Song, and it was really nice talk. I was very interested. I'm now studying stochastic model reduction using quasi-steady state approximation, and also studying about sleep pattern using wearable devices. Thank you.
Yeah, so recently Yumin actually published a work where actually he derived a complete condition under which the deterministic non-elementary function can be used for the Gillespie algorithm. So he,
Oh, fantastic. Okay.
yeah.
Yep.
So actually,
Yeah,
I began that work a couple of years ago, but it was the error analysis was quite approximate, but he did a really complete analysis. Now we know the clear criteria for that. So yeah. I know, you also have interest in this model reduction. So yeah.
Yeah, we thought about it a bit. Often in the context of if you're trying to calibrate a model to data, sort of that can be very kind of costly, in terms of computational simulation. And so we're trying to think about how to use reduced models. So yeah, I'd be really interested to hear about that.
Yeah. And then maybe Hyokyo, you can introduce yourself.
Hi, Professor Kerr. Thank you for the
Hi.
interesting talk. And
Thank you.
I'm Hyokyo Hong, studying under Professor Kim, and I'm also working on Bayesian statistic and inference, and mainly, and other than Bayesian statistics, I'm working on theoretical aspects of stochastic modeling of reaction network. So, actually, I had one question about
yes.
your talk. Yeah, because of urgent calling. Sorry for leaving at some time. So, for the synthetic likelihood from the SDE you compute, I believe that to compute the synthetic likelihood, we have to at least simulate the mean or variance trajectory from the SDE. So how did you do that for the simulation?
So you've got, that's a really good question. So let me hang on, let me try and find the slides, relevant slides. So we've got some different summary statistics, which are, I can't find them right now. It's gone back to the wrong place. Hang on a second. I'll do that. So essentially what you do is that we've got 4 summary statistics. And so each one of those, we, from our data, have, we're going to assume, we can calculate them from our data, and we're going to assume they're normally distributed, some mean and variance, that's what we estimate. And then the idea is that we want to sample parameter values from our prior distribution. We're going to simulate our model multiple times using those parameter values, collect the same summary statistics, and look at the distribution that comes from those summary statistics. So we're assuming those summary statistics are sort of normally distributed. And I guess then that makes it sort of tractable in terms of more tractable in terms of the likelihood, because we're working under the assumption that everything's normally or Gaussian distributed. Does that make sense?
Oh, then you said that you computed summary statistics from the individual realization of the SDE, then they must have consumed lots of computation time.
A lot of time. Yeah, it wasn't too bad. So I think so there are obviously multiple ways we could have done this. We could have done this using, so here's a list of the statistics. We could have done this using a approximate Bayesian computation. But obviously then it's got its own kind of computational limitations. So I don't think there was maybe much in it, but in this context, maybe it was interesting to learn about this. This seemed to be a problem where, because we had quite a lot of data, so we had on the order of between 50 and 55 trajectories for each of the,
Oh.
across the 2 experiments. So we
I
had quite a lot of data. So it seemed that like this was one of those cases where we could apply synthetic Bayes, and it was kind of fun to think about what it would look like. So this, I was going to say, these are the summary statistics, and then we ended up with a long algorithm. And this is because one of the things that we did was we sort of, where's a better slide? We kind of used a sort of synthetic Monte Carlo version of it. Sorry, sequential Monte Carlo version of synthetic likelihoods, where we have a series of intermediate target distributions. So, so a bit like kind of SMC ABC, where we've got a temperature that we evolve from 0 to 1. So if it's 0, it's just the prior. By the time it gets to 1, you're targeting the actual posterior distribution. But this allows you to effectively it kind of, so if you saw like our prior distribution was huge, right? So this allows you to kind of in a sequential fashion essentially target the posterior through a number of intermediate distributions that approximate that posterior. So yeah, it was a little bit, it is time consuming, but I think you save because you have got the likelihood, and also we could make it a bit more in the same way you would use SMC ABC, we could do something similar here, and that was what kind of made it probably doable.
So for the realization, do you use some Gillespie algorithm or tau leaping, or?
No, we just use a fixed time step, just a simulation of the, so like RnMrna, so it's a simulation, right? Solution for the SDE. Yeah, so there's, in a sense, it's not difficult to solve that SDE numerically.
So you directly just solve the SDE.
We don't solve it, but we numerically approximate it using the simplest approach. But there was there are a few things under the hood in the synthetic Bayes that we did to make it, or Tom did to make it a bit quicker.
See, got it. So Hyokyo, is it clear to you?
Yeah. Yes.
Yeah, actually, it's very similar to what you're. Actually, Hyokyo is working on Bayesian inference for non-Markovian system, which has a time delay distribution. So,
okay.
Yeah. So actually, he developed sort of the similar method what you are doing, but
okay.
Yeah, but he's trying to, because due to the time consumption, he tried to derive the analytically the mean and variance from the model.
Okay.
Yeah, so that to avoid the realization, but of course it's not always possible depending on the system. Yeah. So
okay, great. If you have anything written up, so with lots of these things are really interesting. So if you have preprints or papers, I'd be really grateful.
We have a couple of papers, so we will send you. But our work is all for the non-Markovian system, so hopefully that's interesting for you. Yeah.
Yeah, definitely. Thank you.
And then maybe Hoyan, do you want to introduce yourself as well?
Okay, can you hear me?
Yeah.
Yep.
Okay. Hi, Professor, and thank you for a very interesting talk. I'm PhD candidate in KAIST, and I'm studying some partial differentiation, especially some diffusion equation. My major interest is about some heterogeneous diffusion equation, and so actually my group has some great interest in the chemotaxis model, as you showed in the electrotaxis part. So and also I studied about some discrete kinetic model, and that model has some kind of concept of velocity and turning frequency. So I really interested in your electrotaxis model because it contains some velocity and some polarity concept. Okay, I have a simplification about that. So if my understanding is correct, in your electrotaxis model, the velocity part is some kind of deterministic model, but some polarity part has some kind of a noise term, stochastic model. So is there a special reason about that?
I see.
Yeah, that's a good question. So I think we sort of came to thinking about this model, right, by doing a bit of a literature survey and seeing what people had used. So we knew we needed a stochastic model in some sense, and we know that one of the things that's generally attractive about this model is that there's essentially like a persistence, right? There's a memory in terms of the cell velocity, right? So the velocity is certainly changing kind of gradually, right? There's not sort of apart from these spontaneous depolarizations, which are quite quick, right? Then it sort of definitely looks like it's sort of there's definitely, well, number one, there's definitely a persistence. Number two, it's definitely unbiased when there's no field. And then if you look at those direction kind of autocorrelation plots, right? So you can see that over time, there's a sort of gradual decrease in the autocorrelation in terms of the movement direction at one time and the movement direction at another time. So that's definitely sort of consistent with this random walk idea where there's a gradual loss there. And then I think, you know, the kind of, I mean, it's all quite phenomenological, but certainly the potential function that we picked is a potential function. I used to have a plot. Let me just check. There's one in the talk. I used to have a plot in the talk that, and I realized it halfway through I'd taken it out, but what it really shows you is that potential function gives you, there's like a ring, right? Basically a radius 1 in terms of the polarity, and essentially that's where all the mass, right, sits, right? So the quasi-steady distribution has got all its mass on a ring that's kind of radius 1 with some sort of width. And then so essentially what happens is that more or less cells are kind of doing a random walker kind of on that potential function, but they stay on the ring most of the time. And then maybe let me see if I've got another talk where I can show you. But so that was, I guess, my best what am I going to say? My best explanation is that you know that did seem to fit the data relatively well, and it's consistent with like the statistics that we see. Yeah, so this is a kind of autonomous model in terms of their quasi-steady distribution. So cells essentially will try and sit with a polarity of magnitude 1, so their speed is v essentially, and they're moving kind of randomly in any direction over time, and that works quite well. So you can still see spontaneous depolarizations. They still will kind of go towards the origin of event, you know, for short periods of time, but they'll quite quickly come back and to just walk around on this ring. And then the way in which you can visualize what happens with the electric field is by kind of shifting mass on this ring. So you either kind of enlarge its radius or shift it along the axes, or change where the weight is. So why did we choose it? I think it's consistent, you know, at a very high level, maybe a bit more hand wavy than a real argument. It's certainly, when you look at the statistics of the model, they're kind of consistent generally with the idea, but I'm sure there are other models as well, right? So in some senses, I guess when you do modeling, you always have to be a bit wary of the fact that your results and your predictions could be model dependent. And so these are clearly predictions that come from a certain model framework, but I think it does a relatively good job, at least in the electric field free case. Very good question.
Okay, thank you.
So actually, in fact, Hoyan actually working a lot of good work in terms of a heterogeneous diffusion. So actually, you figure out what is the correct way to simulate heterogeneous diffusion system, and then actually he's working with the experimental group to confirm that new model for that system.
Oh, fantastic! Yeah,
that's my research goal, he said.
Yeah. So what is the best way to simulate the heterogeneous diffusion system? So yeah, he doesn't like a current method. So
yes, I'm just using some Monte Carlo method always. So actually, I really I don't know about some kind of a stochastic model because I'm just working on deterministic model. So I'm not sure what is the good simulation, but
for the heterogeneous one, yeah. And then finally, so there are two people, maybe Seho.
Hello, Professor Ruth. Thank you for the nice talk. And I am undergraduate student in Professor Kim's lab, and I'm working on sleep-wake cycle with wearable device, and also I interested in about the method for inferring causality in biological systems.
Yeah, so he has working on developing an algorithm to detect the inference from the time series data set. So yeah, he has developed very nice model-based inference method. Actually, with Danny Forger, and I have tried to study that system for 7 years, but we haven't figured out. But Seo recently solved that mystery. So yeah,
Brilliant.
and yeah, actually he's graduating, and then he's trying to go outside for his graduate study. So hopefully, maybe. Did you apply to Oxford?
Yeah.
I don't know.
Yeah, definitely.
Yeah. And then
Sounds
finally, yeah, Sungmin Ha is our talented undergraduate student.
Hello, Professor Rose.
Hi,
Thank you for the great talk, and I'm also an undergrad student under Professor J, and I am researching on plan circuit and clock, and I also am like having some problem in selecting the model, so it was a really interesting talk. I really enjoyed the talk today, and I'm also working with Seo about the causality research too.
Yeah.
fantastic.
Yeah, actually, yeah. So we have a good collaborator for the plant circadian system. So actually, she measured the various time series data for plant circadian oscillatory time series data, and then actually there are about tens of candidates of the model, actually, which all of them can reproduce that data set well. So we need a way to find what is the best model for that in this situation. So that's why your that model selection method opened our eyes. So certainly after this talk, with Sungmin, we'll go through your paper, and then we will try to use it for our current situation. Yeah.
Yeah, I mean, I can send the link. So it's taking a look, like it's all in the supplementary information.
Yes,
And
great.
so you should be able to, I think, get a good sense of what we've done in terms of weight. So I guess, yeah, that's all in there.
Yeah, so yeah, that would be really, really helpful for us. Yes. Great. Yeah, so before closing the talk, any other question from audiences or other people in this room? I know, yeah, it's passing the dinner time, so.
Yeah, sorry. Thank you thank you all for your time, and thank you for staying back late.
Yeah,
I guess
so Ruth, hopefully, yeah, you can visit Korea after this pandemic. So
love to. That would be really great. Yeah.
So you're more than welcome to visit our research center. So hopefully
thank you. And the same goes in reverse. If anyone wants to come and visit here, they should.
Yes, certainly. Yeah. Yeah, I really look forward to do it. Yeah. So indeed, thanks for giving a talk in such your early time. I know you have a kid, so it was challenging for you. So.
It's okay. Thank you. I think you had to stay late, so I just took them to school and then I came here.
Yeah. And yeah, and everyone, this is the last colloquium of this year. So next week, please don't come. So this is the last one. So yeah. We should thank one more time to the Professor Ruth Baker. Yeah.
Thank you, everyone. Great chatting with you. Thank you for all. And please do send some relevant references. It sounds fantastic.
Yeah, that's great. So let's keep in touch, Ruth. Yeah.
Yeah. Take care.
Yeah. Thank you, Ruth.
Bye.
Yeah. Okay. Yeah. Yeah. 모두들 수고하셨습니다. 네, 그러면 맛있게 식사하시고, 네, 그러면 또 내일은 저널 클럽 있으니까 내일 저널 클럽은 10시입니다. 그래서 모두 10시에 봅시다. 네, 바이바이.